Having examined the various lipids and the nature of their interactions in forming lipid bilayers, let's now consider the strength of those interactions as related to the fluidity of the membrane. Owing to the varied nature of the lipid tails embedded in the membrane, the hydrocarbon interior of the bilayer may be more or less ordered. In other words, they may be ordered and rigid, or disordered and more fluid. As we saw in a previous lesson, saturated fatty acid chains are linear in geometry, as shown on the left below. This leads to the maximum number of van der Waals interactions and therefore a more rigid arrangement. In contrast, unsaturated fatty acids have one or more kinks in their chains, disrupting some of the van der Waals forces. Note that a monounsaturated chain like the one in the center of our figure is less kinked than a polyunsaturated chain like the one on the right. This unsaturation leads to weaker intermolecular forces and therefore greater fluidity of movement. The more unsaturated the chain, the more disordered and fluid the lipids are within the membrane. The presence of cholesterol can also influence the fluidity of membranes. As considered previously, and as illustrated here, the fused ring structure of cholesterol is very rigid. It serves to stabilize the straight chain arrangement of the fatty acid tails. This increases the effect of the van der Waals interactions and leads to a more rigid membrane structure. How does membrane fluidity compare and contrast between plants and animals? It may not be apparent, but animal membranes are actually more rigid than those of plants. The latter have a higher percentage of unsaturated fatty acids and therefore more fluid membrane structures. Also, plants lack cholesterol and its ordering effect. If you're thinking of the rigid structure of plants overall, keep in mind that we are discussing just the membranes here and not the rigid structure of the plant cell wall. Prokaryotes have the most fluid membranes, not possessing any appreciable amount of steroids. Another factor that influences membrane fluidity is the presence of heat. The lipids become more fluid in the presence of heat, and the temperature at which this transition occurs has to do with the types of lipids present. Membranes containing more ordered lipids, and therefore stronger van der Waals interactions, have a higher transition temperature, called the TM, or melting temperature. If we examine the structural changes associated with temperature elevation, we find, as illustrated below, that the thickness of the membrane decreases and the surface area increases. This is an important consideration for animals that experience variability of temperature and whose membrane integrity might be compromised as a result.